Clarity 301 has one pre-specified interim efficacy analysis. It was written into the statistical analysis plan before the first patient enrolled. The DSMB knows when it happens. FDA knows when it happens. I know when it happens. Meyer Osei, Arventa Global, Clinical Trial Manager. The interim analysis occurs when we reach 208 progression-free survival events, 65% of the 320 target events for the primary analysis. At that point, the independent statistician unblocks the data for the DSMB. The DSMB reviews the unblinded hazard ratio and compares it to the pre-specified efficacy boundary. The efficacy boundary at interim is more stringent than at final. That's alpha spending. We're borrowing statistical significance from the final analysis to look early. If we use alpha at the interim, we have less alpha left at the final. The boundary is O'Brien-Fleming, very conservative at interim, nearly identical to the final analysis threshold at final. 3 outcomes. The hazard ratio crosses the efficacy boundary. The DSMB recommends stopping for overwhelming efficacy. It crosses the futility boundary. The DSMB recommends stopping because the drug isn't working. Or it doesn't cross either boundary. The trial continues to full enrollment. Operationally, interim analysis is an event-driven milestone. I'm tracking PFS event count in real time. At approximately 200 events, operations intensify. Data cleaning milestones accelerate. The database is not locked. We're not there yet, but we're cleaning toward it. Agentforce event count monitoring surfaces, PFS event, accrual across sites in real time. Pre-specified interim analysis, the statistical and regulatory framework. An interim analysis is a planned evaluation of accumulating trial data before the pre-specified endpoint for the primary analysis is reached. For a survival or time-to-event endpoint like PFS, the interim analysis is typically defined in terms of the number of events, disease progression or death, that have occurred rather than calendar time. Because the statistical information in a survival trial is proportional to the number of events, not the amount of time elapsed. In Clarity 301, the interim analysis is pre-specified to occur at 208 PFS events, 65% of the 320 event target for the primary analysis. The pre-specification is critical. If the timing or threshold of an interim analysis is determined after the trial has started, and especially after any unblinded information about the data has been seen, the analysis is post hoc and loses its statistical validity. FDA requires that interim analyses be pre-specified in the statistical analysis plan, SAP, filed with the IND before the analysis occurs. Unplanned or post-hoc interim analyses are viewed with significant regulatory suspicion. Alpha spending, the statistical cost of looking early. The fundamental statistical problem with interim analyses is that looking at accumulating data multiple times inflates the type 1 error rate, the probability of incorrectly concluding that the drug works when it does not. If you apply the conventional P0.05 threshold at both an interim and a final analysis, the overall probability of a false positive result across both looks is approximately 3%, not 5%. Alpha spending functions are mathematical methods for distributing the available type 1 error, alpha equals 0.05 for Clarity 301, across the planned interim and final analyses, such that the total type 1 error rate across all looks does not exceed 0.05. The O'Brien-Fleming alpha spending function, one of the most widely used in Phase 3 oncology trials, allocates very little alpha to early interim analyses, requiring a very large treatment effect to stop early for efficacy, and preserves most of the alpha for the primary analysis, where the threshold is nearly identical to what it would be without any interim analysis. The practical implication: to stop Clarity 301 for overwhelming efficacy at the interim analysis, the hazard ratio for PFS would need to be substantially more impressive than the 0.75 hazard ratio, P0.05, with O'Brien-Fleming adjustment, required at the primary analysis. Futility boundaries, protecting patients from an ineffective therapy. In addition to efficacy stopping rules, stop early because the drug is working dramatically well, the DSMB charter specifies futility stopping rules. Stop early because the trial is very unlikely to demonstrate efficacy even if enrollment continues to completion. Futility analysis is a form of conditional power calculation. Given the data observed so far, what is the probability that the trial will achieve a statistically significant result at the primary analysis? If the conditional power is below a pre-specified threshold, commonly 10–20%, the DSMB may recommend stopping for futility. Futility stopping protects trial participants in the control arm from continued randomization to a drug that is unlikely to benefit them, and protects the sponsor from the cost of completing a trial that will not achieve its primary endpoint. Futility stopping rules can be binding. The trial must stop if the boundary is crossed, or non-binding. The DSMB may recommend stopping, but the recommendation is advisory. In Clarity 301, the futility boundary is non-binding. The DSMB can recommend stopping, but Arventa's leadership must formally accept the recommendation. Event-driven design, operational implications. In a PFS endpoint Phase 3 trial, the primary analysis occurs when a pre-specified number of progression events have been observed across all enrolled patients, not at a calendar date. This event-driven design creates operational complexity for the CTM. Enrollment can be complete, all 312 patients randomized, but the primary analysis cannot occur until 320 patients have had a documented progression event or died. The time from last patient enrolled to the primary analysis event threshold is the follow-up period. Patients continue on study treatment or in follow-up, and progression assessments continue according to the protocol schedule. For Clarity 301, with a median PFS estimated at approximately 8 months in the experimental arm and 4 months in the control arm. Docetaxel. Most events will occur relatively quickly after enrollment, but some patients with long PFS on veliratinib will require extended follow-up before the event target is reached. The CTM monitors the event accrual curve, the rate at which PFS events are occurring across the enrolled population, to project when the interim and primary analysis thresholds will be reached, and to plan the data management activities, accelerated data cleaning, central laboratory reconciliation, imaging data lock, that must occur before the analysis. Adaptive design elements, planned flexibility within pre-specified boundaries. Clarity 301 is not a complex adaptive trial. It does not have pre-specified sample size re-estimation or adaptive randomization. However, it incorporates some adaptive elements within the pre-specified design. The enrollment stop rule: enrollment stops automatically when 312 patients are randomized, regardless of the calendar date, and the event-driven analysis trigger. The primary analysis is initiated by reaching 320 events, not by a calendar date. More complex adaptive designs, response adaptive randomization, seamless Phase 2/Roman 3 designs, master protocols with multiple arms and shared control, are increasingly common in oncology development but require specific FDA interaction, adaptive design guidance, pre-submission meetings, and more complex statistical infrastructure than traditional Phase 3 designs. The statistical analysis plan for Clarity 301 specifies that all adaptations are pre-planned and that no unblinded data will be used to modify the trial design after initiation, a commitment that preserves the Type 1 error control that FDA requires for a confirmatory trial. Database lock day is one of the most significant milestones in a Phase 3 trial. After lock, no data changes. The statistical analysis plan executes on the frozen data. What we submit to FDA is what was in that database on that day. Meyer Osei, Arventa Global, Clinical Trial Manager. The lock process for Clarity 301 took 11 weeks. It is not a single action, it is a sequence of interdependent closures. Imaging data, lock first. The Independent Imaging Review Facility finalizes all radiological reads. Central Laboratory Reconciliation. Every lab value in the EDC is confirmed against the central lab's own records. SAE Reconciliation. Every serious adverse event in the pharmacovigilance database is confirmed against the EDC. Medical Coding Review. Every adverse event term is mapped to the correct med-DRA preferred term and system organ class. When all data streams are reconciled and all outstanding queries are resolved, the data manager performs a final quality check. The biostatistics team signs off. The medical monitor signs off. I sign off as clinical operations lead. The EDC vendor executes the lock. The database is now read-only. The clinical study report, the CSR, documents the entire trial. Design, conduct, patient population, results, and conclusions. It is co-authored by clinical, biostatistics, and medical writing. It is the primary deliverable from this trial and forms the backbone of the NDA. Agentforce lock readiness tracking, query resolution rates, reconciliation status, milestone completion across all 45 sites. Database lock, the formal freeze of trial data. Database lock is the formal action that closes the trial database to further data entry, query resolution, or modification. After database lock, the data in the electronic data capture system is fixed. It becomes the dataset on which the statistical analysis plan is executed and the results that will be submitted to FDA. The lock is irreversible in normal operations. Any data changes after lock require a database unlock, which is an auditable event that must be documented and justified. For Clarity 301, the database lock follows a defined sequence. Imaging data lock, independent imaging review facility, finalizes all radiological assessments of tumor response and progression. Central laboratory reconciliation. All lab values in the EDC are cross-checked against the central laboratory's records to confirm accurate transcription. SAE reconciliation. All SAEs in the EDC are cross-checked against the pharmacovigilance database to confirm that every serious adverse event is captured in both systems. Medical coding review. All adverse event terms and medical history terms are coded to the current med DRA dictionary, and all concomitant medications are coded to the WHO drug dictionary. And final data review. The data management team, medical monitor, and biostatistics team review the complete dataset for any remaining issues. Only when all these steps are complete and confirmed is the database locked. Audit trail, the regulatory record of data integrity. Every action performed in the EDC, data entry, data modification, query creation, query response, user login, database lock, is captured in an immutable audit trail that records who performed the action, when, what the previous value was, and what the new value is. The audit trail cannot be deleted, modified, or hidden. It is a regulatory requirement under 21 CFR Part 11, Electronic Records and Electronic Signatures, and IC E6 GCP. FDA inspectors reviewing an NDA submission will examine the audit trail to verify that data has not been improperly altered. Specifically, they look for patterns such as data that was modified after an investigator became aware of a regulatory inspection, data that was modified to correct a value after the unblinded results became known, or data modifications that lack adequate documentation. A clean audit trail, one that shows only expected data modifications, correcting transcription errors with explanations, closing queries with appropriate documentation, is a sign of a well-managed trial. An audit trail with unexplained bulk modifications, modifications by unauthorized users, or modifications that occur at unusual times, is a significant inspection, finding. Medical coding, standardizing adverse event terminology. Before database lock, every adverse event and medical history term collected in the trial must be coded to a standardized dictionary to enable consistent analysis and regulatory review. The MedDraw Medical Dictionary for Regulatory Activities dictionary is the international standard for coding adverse event terms in clinical trials. The WHO Drug Dictionary is used for coding concomitant medications. Medical coding is performed by trained medical coders who map the verbatim term entered by the site coordinator. Nausea and vomiting, joint pain, decreased appetite, to the appropriate med DRA preferred term and system organ class. Accurate coding is critical for the adverse event summary tables in the clinical study report, tables that are reviewed by FDA and are the basis for the safety sections of the drug label. Miscoding, mapping a verbatim term to the wrong preferred term, can systematically misrepresent the safety profile of the drug, making a common toxicity appear rare or distributing toxicity across multiple system organ classes that should be grouped together. Clinical Study Report, the Comprehensive Trial Narrative. The Clinical Study Report, CSR, is the document that comprehensively describes the design, conduct, analysis, and results of Clarity 301. It is the primary scientific and regulatory deliverable from the trial, forming the backbone of Module 5 of the NDA submission. The ICH E3 guideline specifies the structure and content requirements for a CSR. The study synopsis, a structured summary of the trial design and results. The introduction, the clinical and scientific background for the study objectives and endpoints, the investigational plan, protocol design, patient population, statistical methods, the study patients, disposition, protocol deviations, baseline characteristics, the efficacy results, primary and secondary endpoint analyses, the safety results, adverse events, laboratory abnormalities, vital signs, discontinuations, the discussion and overall conclusions, and a series of appendices, protocol, SAP, investigator listing, patient listings. The CSR for a Phase 3 oncology trial, like Clarity 301, typically runs to several thousand pages, including appendices. The main body is 200–400 pages. Co-authorship involves clinical, biostatistics, medical writing, and regulatory affairs, with the CTM responsible for the study conduct sections describing site operations, monitoring activities, and protocol deviations, CSR quality, and the NDA submission. The CSR is not merely an internal document. It is a regulatory submission that FDA reviewers will read in detail, and discrepancies between the CSR and the NDA submission dataset, the actual data files submitted electronically, are a serious regulatory deficiency. The data in the CSR summary tables must match the data in the electronic datasets. The patient counts must be consistent across all sections. The adverse event tables must use consistent denominators. The protocol deviation listings must match the deviation database. Pre-NDA submission, the sponsor conducts a comprehensive quality check of the CSR against the submission datasets. A process called data verification, or integrated quality review, to ensure internal consistency before the package reaches FDA reviewers. A CSR with numerical inconsistencies, patient count discrepancies, or findings that are described differently in the results section. And the discussion section will generate FDA information requests that delay review and can damage the credibility of the submission. The statistical analysis plan for Clarity 301 was finalized before the first patient enrolled, before we saw a single data point. That's not a technicality, it is the foundation of regulatory credibility. Meyer Osei, Arventa Global, Clinical Trial Manager. The SAP defines every analysis that will be run on the Clarity 301 data. Primary analysis, progression-free survival in the intent-to-treat population, assessed by the log-rank test, stratified by prior lines of therapy and geographic region. Hazard ratio estimated by Cox proportional hazards model. Secondary endpoints are tested in a pre-specified hierarchical order. Overall survival first, then confirmed objective response rate, then duration of response. Hierarchical testing controls the family-wise error rate. We can only claim a secondary endpoint is statistically significant if the endpoint before it in the hierarchy was also statistically significant. This prevents cherry picking. The SAP also specifies sensitivity analyses, alternative approaches to the primary analysis that test the robustness of the primary result. What happens if we censor patients who start a subsequent therapy? What happens if we use a different censoring rule for patients lost to follow-up? If the primary and sensitivity analyses point the same direction, the conclusion is robust. If they diverge, FDA wants an explanation. The SAP is pre-registered with FDA as part of the SBA agreement. After database lock, biostatistics executes the SAP exactly as written. Agentforce biostatistics milestone tracking, SAP version control, analysis execution status, table shell review, statistical analysis plan, structure and regulatory purpose. The statistical analysis plan, SAP, is the comprehensive document that pre-specifies all statistical analyses to be performed on the clinical trial data. For a Phase 3 confirmatory trial like Clarity 301, the SAP is the biostatistics team's commitment, made before the data are unblinded, to a specific set of analyses, using specific statistical methods, applied to specific patient populations, with specific handling rules for missing data, protocol deviations, and censoring. The pre-specification of analyses before data unblinding is the cornerstone of confirmatory trial statistics. If the sponsor could choose which analysis to run after seeing the data, the primary endpoint population, the statistical test, the covariates in the regression model, the censoring rules for events that don't clearly meet the endpoint definition, they could select the analysis that produces the most favorable result, inflating the apparent statistical significance of the trial outcome. FDA requires that the SAP be finalized, with a dated version-controlled signature page, before the database is locked and the unblinded analysis is conducted. A SAP amendment made after any unblinded data has been seen by the sponsor is treated as a post-hoc analysis, regardless of the stated rationale. Multiplicity and hierarchical testing, controlling the false positive rate. A Phase 3 trial typically tests one primary endpoint and multiple secondary endpoints. Testing multiple hypotheses simultaneously inflates the type 1 error rate. If you test 5 endpoints each at p0, 0.05, the probability of at least one false positive result is approximately 23%, not 5%. Hierarchical testing is the pre-specified strategy for controlling the family-wise error rate across the primary and secondary endpoints in Clarity 301. The endpoints are ranked in order of clinical importance: PFS, primary; overall survival, first secondary; confirmed objective response rate, second secondary; duration of response, third secondary. Statistical significance for each endpoint is declared only if all preceding endpoints in the hierarchy were also statistically significant. If PFS meets the primary endpoint threshold but overall survival does not, the objective response rate and duration of response cannot be claimed as statistically significant. They can be reported descriptively, but not used to support labeling claims. This hierarchical structure is agreed with FDA before the trial begins and is specified in the SAP. It prevents post-hoc selection of which secondary endpoints to emphasize based on the data. Sensitivity analyses, testing the robustness of the primary result. Sensitivity analyses are alternative analyses of the primary endpoint that use different assumptions, different patient populations, or different statistical methods to assess whether the primary conclusion is robust to methodological choices. In Clarity 301, pre-specified sensitivity analyses for the primary PFS endpoint include a per-protocol analysis, restricting to patients who met all eligibility criteria, received study treatment, and had primary endpoint assessments within protocol windows, excluding patients with major protocol deviations that affect the primary endpoint. An alternative censoring analysis, using a different rule for patients who start a subsequent anti-cancer therapy before progression is documented. Should they be censored at the start of the subsequent therapy or at the date of the assessment that would have identified progression? And a landmark analysis, assessing PFS outcomes at a fixed time point, the proportion of patients who are progression-free at 12 months. If the primary analysis and all sensitivity analyses show a consistent, statistically significant improvement in PFS for velorafenib versus docetaxel, FDA can be confident that the result is not an artifact of a particular methodological choice. Divergence between the primary and sensitivity analyses requires explanation and may generate FDA information requests. Analysis populations, IT, per protocol, and safety. The SAP defines the patient populations that will be used for each analysis. The intent-to-treat IT population includes all randomized patients, regardless of whether they received study treatment, complied with the protocol, or had evaluable primary endpoint data. ITT analysis is generally considered the most conservative and least susceptible to bias, because exclusion of any patients from the primary analysis population, even for documented reasons like protocol deviations, creates potential for bias if the excluded patients are systematically different between arms. The per-protocol, PP population includes only patients who were eligible, received study treatment, and had primary endpoint assessments according to the protocol. It excludes patients with major protocol deviations that affect the primary endpoint assessment. The safety population includes all patients who received at least one dose of study treatment and is the primary denominator for adverse event analyses. For Clarity 301, the primary efficacy analysis is the ITT population. The PP analysis is a pre-specified sensitivity analysis. The safety analysis uses the safety population. The relationship between ITT and PP results is informative. If the ITT and PP analyses give similar results, protocol deviations are unlikely to have affected the primary conclusion. Pre-registration and the SBA commitment. The SAP for Clarity 301 is linked to the Special Protocol Assessment (SPA) agreement with FDA. The formal agreement that the trial design and statistical analysis approach are adequate to support a new drug application if the primary endpoint is met. The SBA commits FDA to honor the agreed statistical analysis plan as the basis for regulatory review, provided the trial is conducted as agreed. In practice, this means that Arventa's biostatistics team cannot change the primary analysis method, the primary endpoint, the key efficacy population, or the alpha level after the SBA is signed without potentially invalidating the SBA agreement and requiring a new Type B FDA meeting. SAP amendments after SBA agreement require FDA notification and, depending on the magnitude of the change, may trigger FDA feedback that affects the SBA status. The SAP is version-controlled, with each version dated and signed. The version that governs the primary analysis is the one finalized immediately before database lock.